Hi, my name is Jennifer Coughlin, and I am the president of the NR2F1 Foundation. And I'm here with Dr. Clement Chow, Professor of Human Genetics at the University of Utah. As the NR2F1 Foundation, at the end of 2024, we focused all our fundraising efforts on a project that we're really excited about, a drug repurposing study using fruit flies. Dr. Chow, can you tell me what exactly is drug repurposing and why is it important for finding new treatments?
Thanks, Jennifer. It's really great to be here with you and kind of to speak to the bigger NR2F1 community. So drug repurposing is, I think, a really important approach for new therapeutic discovery, especially in the rare disease world. As many of you know, rare disease drug discovery is almost non-existent, and it's very difficult because it's time-consuming and also very expensive. And drug repurposing kind of fills that gap by simply asking, are there drugs that are already FDA approved that might be able to solve our problems for us, right? Can we find new uses for old drugs? And it's important because many of you have also had drugs that have been prescribed off-label for a purpose they were not originally approved for through the FDA. And that's totally normal and okay. And so if we can find a reasonable drug that can work for a rare disease for NR2F1 individuals, then it might move to the clinic quite a bit faster than kind of a novel drug discovery.
Why did you choose to use fruit flies in your research? What makes them a good model for studying human disease?
Yeah, so fruit flies really kind of fit in this, I think, really interesting nexus amongst all the model organisms that we can choose from. It's small and has a very short generation time, so it's only 10 days from generation to generation. And so that means that we can generate as many flies as possible for very little money, and it takes very little amount of space to do a lot of work. And and so those are kind of some of the advantages that make it a really great model in general. But for drug diseases, for discovering drugs for new diseases, it's a good model because everyone wants a drug really fast. And we can take advantage of these the short lifespan and the economy of the fly to look for these drugs. And the flies are also they're very similar to humans in their physiology. It may not seem like it on the surface, but in fact, they have to ingest the drug just like humans. It has to go through their digestive tract, go through a circulation, get through a blood-brain barrier before it can have its effect in the brain or elsewhere. And so for those reasons, I think we really love using the fly for this kind of work.
So when you give your flies BBSOAS, can you actually see it in them? Do they look different?
Yeah, so just to kind of give you just an idea of how we decide what we're looking for in a model that we use for a drug screen. We either generate the mutation or generate a fly that's very similar to the situation we see in the patients and in the kids, and we ask what's going wrong when we do this in the fly. And when we eliminate NR2F1 function in the fly, we get a number of different phenotypes that we can observe. And but we can study all of those as a model of NR2F1 dysfunction, but for a drug screen, we really need the kind of phenotype that's really well separated from what the kind of wild type control condition is. And so we have a lot of experience with these drug screens, and we basically just run through the list of phenotypes that we're really good at screening on, and we pick the best one that we can find for that gene. And in the case of NR2F1, it's actually the wing, and I'm going to show that to you right now. So here in this figure, this is basically the basis of the drug screen. Here on the left is a control healthy fly, has full NR2F1 function. And if we eliminate NR2F1 function in the wing of the fly, we get this really abnormal looking wing. You don't have to be a scientist to see that there's a
That's
quite big,
amazing.
yeah. You don't have to be a scientist to see that there's quite a big difference here. And and we basically systematically did this for different body parts and whole flies, and kind of found that the wing was the best case scenario for a drug screen. And so we'll take these flies on the right with these abnormal wings, and give them drugs and simply ask which drugs can move this abnormal wing closer to what we see in a healthy wild type fly. So we're trying to correct this abnormality and make it look more healthy. And it's a really simple assay, and the simpler these assays are, the more successful the screens tend to be.
And how do you know if a drug that works on fruit flies will also work in humans?
Yeah, that's a really good question, and the simple answer is that we don't. Um, but we can kind of lean on a couple facts, in that the biology for these for cells are mostly the same, evolutionary conserved between flies and humans. The function of NR2F1 is is the same between flies and humans, and so while humans don't have wings, the wing is just the output of the dysfunction in the cell that we're doing, right? And so each organism has a different output, but the kind of core problem is the same. And so we're hoping that we see an effect in flies, we'll see an effect in humans. But as with most things in science, we don't know until we try a lot of times.
I think as a parent, is for us, it very much feels like it's always worth the try.
Yeah, yeah, I can understand that.
And what are the potential benefits of the repurposing for families with rare diseases like BBSOAS?
Well, the kind of benefit is, of course, that there could be a drug kind of hiding in plain sight, right, for the community. And kind of that's the big kind of best case scenario benefit, right? But the kind of other benefits is that it's a faster route, right, to the clinic. And so even if we find a number of drugs that maybe aren't a slam dunk right away, we immediately have a number of options that we can develop really quickly. And that's a much faster process than going from like a novel new drug discovery. Um,
Mhm.
and so I think that we've had screens in the past that have gone straight from the drug screen in the flies to patients within weeks. And we've had screens where we've found a number of hits that make sense, but maybe it's not a slam dunk that we give these to kids. And so there's a little bit more development. And we've had screens where we find drugs that need a little bit more kind of biology before we make a decision. But any of those scenarios are really great because, for most rare diseases, there's nothing, right?
Yeah.
And so we go from nothing to a pretty good lead pretty quickly with this type of approach.
hmm That's why we are definitely, as a community, so excited about it. And in terms of the next steps for this project, from where you are right now, what's on the timeline for you?
Yeah, so this project is just getting started now. After conversations with Jen and the foundation, we did a little bit of work to develop the model that I just showed you. We kind of worked out the best case conditions for this drug screen, and we're ready to go. So I think that my team will start this drug screen pretty quickly, and once it gets going, it'll be about 4 months to screen through about 1,500 FDA approved drugs. And then from there, we will have some more discussions with the foundation about what the best next steps are. And then from there, we can kind of discuss what we'll do next. But it's a couple months.
That's fantastic. And for families who are watching, how can families get involved with this?
I think the I think the best way the families can help with at least this type of work is really to kind of be involved in the natural history studies and kind of be involved in kind of helping the foundation collect the data that they need. Um, it may be up, we may find a drug that's, you know, off the shelf, Advil, for example, and it may be a miracle drug. It may not be. We may find a drug that requires prescription or pretty careful dosing, and in any of those cases, we really need to understand, what the natural history is and what kind. And as the clinicians kind of start monitoring the use of a new drug, they really need to know kind of what biomarkers to look for, what changes we should expect to see in the patients. And so the more data that the foundation collect in a natural history survey or study is really helpful. It's a lot of work. I know it's a lot of work for families to do this, but it's really worth it in the end. Even for and it'll be worth it down the road too for even more complex therapies. If gene therapy is sometime down the road, you'll really need it for that. So
Oh,
it's always kind of keep working with the foundation and try your best to kind of get as much information as possible to them.
okay. So families need to go and complete the NR2F1 patient registry because it's
Yes.
critical that we have a history of each of the patient's symptoms and experience. Because if
Yeah,
we get to potential drug trials, then we need to be able to measure and see and show the positive impact essentially in the data.
yeah, that's
Um,
exactly it.
Great. So, for everyone listening, if you haven't already, please go and sign up to the NR2F1 patient registry and complete all the surveys, because this will mean that you're ready to participate when a study comes around. Dr. Chow, thank you so much for sharing with us and helping us as families better understand the work that you're doing. And we wish you all the best of luck for the next 4 months. And we can't wait to hear. And hopefully, we can come back for a second interview once you have the results and think about what's next. But in the meantime, thank you for all the support for the NR2F1 Foundation and those living with BBSOAS.
Yeah, thank you for speaking with me. We're really excited to work with you and looking forward to sharing whatever we find.
Thanks so much. We'll talk to you soon.
Bye-bye.